So, this is the apple extract, we have taken it in a Falcon tube. Here we can see this is a deep brown color. Okay. So, we are not going to use this entire solution for our analysis, we are going to dilute it. Basically, we are going to make it half dilute. Okay. For that, what we are going to make is that we are going to take another clean Falcon, okay, and we are going to pour around 20 ml of 20 to 25 ml of this apple extract. And the remaining we are going to dilute it with double distilled water. So, this color appears much more soothing as compared to the previous one. So, this straw yellow colored, or rather a bit brownish yellow color solution we are going to use for our analysis. Now, let us proceed to the enzyme kinetics study. So, now let us start with the kinetics experiment. For that, we will be using a UV spectrophotometer. In the previous experimental biochemistry course, we have actually seen how to use this UV spectrophotometer, what are its function, about its compartment, and etc. All those things we have actually have seen there. Well, we have prepared the solution starting from catechol and inhibitors along with the enzyme extract, which is the apple extract, and now we will shift to the experimental part. As you can see here, this one is actually the UV instrument, and the UV spectrophotometer out here is similar to that we have used earlier, but basically this is a different type of instrument out here. Now, the compartment and about the instrumental parameters and how to set up this instrument, how to use this instrument, we have seen in the previous experimental classes. Pretty same are there in the software also. Now, what we can see out here in the software tool, this is the screen. Here we can see that this axis is the absorbance one, and this one is the nanometer wavelength part. Now, for kinetic study, what we need to do is that we need to shift this one from the absorbance versus wavelength to absorbance versus time. For that, how we can do is that we should go to window and select. From here we can see kinetics, photometric and spectrum. Okay. From here we can actually select this kinetics. Once we select this kinetics, we can see there is a change in this appearance of the software in the software screen out here, and here we can see this is absorbance part, and here in the time, and the time is there in seconds. Before beginning with this experiment, we need to connect this one as we have seen in the previous UV spectrophotometer also. If we press connect out here, all those other parameters will get enabled. Now, one important thing is there, we need to get the parameters ready. Now, for this parameter we need to click this one M. This is similar to the previous UV spectrophotometer which we have used, this method 1 M, and here it is the kinetic mode is showing, this is a kinetic method. Various options are there like measurements, sample preparation, file, etc. Let us see one by one what we get here. Now, here we can see in the timing mode, one is given in auto and the other is given manual. So, in manual part, what we can see is that the units basically we can change from second, minute to hour. However, for doing kinetics experiment in biochemistry, we have to generally stick to the second part. The next thing is the total time. However, the total time is taking 300. Why? Because of the cycle time and a number of readings. So, this are basically if you take into account these two things, you can land up to total time 300 seconds, and these are the activity region and 20 to 40. However, we are not going into manual part, we will stick to auto part, and the total time out here will be 5 minutes, which is basically 300 seconds. So, here the units are second and total time is 300. And if we are not bothering with the cycle time and number of readings out here, and the activated region is 20 and 40. This is 20 second to 40 second. These are by default, but however, you can change once you start with the experiment. We will show you later on. Next thing we need to see here the wavelength. The wavelength given in nanometer. The number of wavelengths we need to study here either single or multiple. Here WL1 minus WL2 or WL1 by WL2, other modes are also shown. However, we are interested with single wavelength, and the wavelength we are going to study is not 290, but 540, because we are going to see the effect on catechol using catechol enzyme, that is the enzyme which catechol oxidase enzyme which actually oxidizes catechol. And all other parameters we did not need to worry about. The other is the sample preparation, and these are things which we do not require out here. File options, which file we should save. So, it is pretty simple and it is same as that the previous one in the spectrum file. We can select here. We are here in this NPTEL folder, as you can see, and week 8 enzyme kinetics course, and we are giving the file name as KIN, or basically kinetics. These are for your self convenience, and we are clicking OK. Now, here one thing we can see here, this is the absorbance we are showing, and we can change here from 0, and this one we can change to 1, and the time here we can see it is starting from 0 and it is going up to 300 seconds. Now, before beginning with this experiment, what we can say is that we need cuvette. So, these are basically cuvettes. The cuvettes are quite familiar. We have seen in the previous biochemistry experimental classes, where the cuvette here one side is actually clean and the other side is actually not clear and not transparent. Okay. And this cuvette we are going to use, this is actually 600 microliter cuvette. So, initially what we need to do, we need to get phosphate buffer filled in this cuvette before beginning with our experiment. Now, we will be using the phosphate buffer. The phosphate buffer we will be using is pH 7.4, 10 millimolar phosphate buffer. So, we are taken this micropipette out here, and this is the cuvette. Okay. Now, I am taking out phosphate buffer, that is 600 microliter from here, and gradually adding this in the cuvette. This whole procedure of how to add the solution in the cuvette you have seen in the previous classes. So, I am not going into details out here. Now, in the UV compartment, you know there were two chambers, one is the control chamber and other is the sample chamber. So, this one what we are going to do is that we are going to take a clean tissue paper and we are going to rub this clear side out here properly and open this lid and transfer it into the control compartment. Okay. In a similar fashion, you are going to take 600 microliter of phosphate buffer out here, clean these two sides properly, and then transfer it into the other compartment, that is the sample compartment, the sample holder basically. Okay. So, the inner part of this UV compartment is pretty same to that of the previous instrument. There are two holders, one is for control and the other is for sample. We are closing this thing and moving on to the screen. Okay, now, we have added this two buffer into the cuvette and placed it in the compartment. Now, we will go to do this baseline correction. For that, we have taken the phosphate buffer in the cuvette and placed it in the compartment, UV compartment. Out here, we will do this baseline, and the baseline here is showing the start is actually 550 and the end is 530. And because it is scanning in the two regions, that is 550, 530, and however, our point of interest is actually 540. So, now for that let us do the baseline correction. Okay. So, we have set this our wavelength out here at 540. The scan will be taking place at this wavelength only. Now, the second thing we need to require is that the addition of substrate and the protein. So, this is a bit a crucial step, and let us see how to carry out this process. Now, we will adjust the concentration of substrate and enzyme within this mixture. In the beginning, we have to add the substrate, that is our catechol. So, the concentration of catechol we are using out here is actually 1 millimolar, and for that we require around 30 microliter of catechol. Okay. And the protein we are adding, or rather the enzyme out here, that is the apple extract we are adding, is 150. So, apple enzyme is actually 150 and our substrate, that is catechol, is 30. So, basically we have to pipette out 150 plus 30, that is 180 microliter of buffer from here. For that we have taken this micro pipette and we have adjusted the volume to up to 180 microliter. We are taking out carefully from here this buffer, 180 microliter buffer we are taking out. Okay. In the beginning, we will be adding 30 microliter of our catechol solution. So, I have taken this micropipette and a fresh tip. Okay. So, this is the catechol solution. The catechol solution we have taken out is this one. So, we are pipetting out this catechol from here, 30 microliter of catechol, and we are adding here in this solution, and we are mixing it after addition. Now, after that what you need to do is that you need to add the protein. Always use a fresh tip for the new solution or new enzyme extract. Now, the protein we are going to take is actually 150 microliter. We have adjusted this micropipette to 150 microliter. We are taking a new tip from this tip box, and this is the protein solution as you have seen. Now, one important thing to remember is that once you take this protein solution, add it immediately in this mixture and immediately start the experiment. Please do not give any time delay for this experiment after or once you have added the protein to it. So, now we have taken this protein solution from here. Okay. We are opening this lid initially, and here we are taking this cuvette, putting this tip and releasing the solution, and up and down a few times and immediately transferring it here, closing this and start. It takes some time for the absorbance to show up in the screen. Here we can see the with change in time an absorbance value is showing, but this is not actually the correct absorbance. We will get the value once the experiment is done with. So, here we can see actually the rate is gradually here increasing, the reaction is proceeding basically. this appears to be somewhat a parabolic, where it gradually increases, but the increase is not that steep, or rather a smooth increase. What happens is that the reaction is actually taking place, and this catechol is actually being oxidized in presence of enzyme, and as a result the absorbance at 540 is changing, which is reflected from this curve. With time the absorbance is changing. So, almost we are done with 250 seconds. And 50 seconds more for the reaction. Now, we can see here it is becoming more or less smooth out here. Initially there is an increase and it is becoming a bit smooth out here. Now, there is an increase in this absorbance value with time, and we will get a result after this experiment. And 297 seconds, 98 and 300 seconds. So, here are certain options or other clue which we can click. Okay. So, what we can see is that when we click this option out here, the main table actually, here it shows some values, and the value which we should be actually bothered is that the activity given here or the milli-absorbance per minute. Okay. So, these two values are actually the thing which you should be bothered, because they are actually what they represent is that they represent the change in the absorbance with time. Okay. And the change is actually over this range. Okay. These two are showing this 20 to 40 20 second to 40 second. Over this range this change is shown. So, milli-absorbance per minute it is being shown. So, what you can do is that initially you need to save this one. Okay. For saving you go to file, save as, I am giving it sample FAM1 underscore catechol. So, this is the first sample for catechol. The saving type is in case of time course .kin kinetics file mode, and again we can save this as a text file, that is a data printable. Whether you save or not save this one, this is not important. However, what you need to do, you need to record the time. So, what I will do is that I will record both the activity, okay, and the milli-absorbance per minute. So, the serial number is 1 and the activity I am getting here is 0.0576, and the milli-absorbance per minute out here we are getting is 57.6092. Okay. So, I am recording this one. One important feature from this curve, as we can see, is that this is somewhat a parabolic, and it is not similar to that of the curve in case of Michaelis-Menten equation, although both are not the same, because in that case it was velocity versus substrate concentration. Out here it is absorbance versus time. Still it appears similar. However, here we can see the absorbance changes with time, which we can see from this type of curved graph. Now, let us shift to the second sample, where we will be using a higher concentration of CCMP. Now we are going to use 2 millimolar of catechol. For that we require 60 microliter of catechol and the total volume is 600 microliter, and the buffer we require is 390 microliter, and the enzyme extract, the apple extract, is actually 150 microliter. Now first let us use this phosphate buffer. Initially we will be taking 390 microliter of phosphate buffer. This is the phosphate buffer, we are taking 390 microliter. Now one important thing is that after you have used this cuvette in the previous for previous sample, you have to clean it properly, so that you have to be sure that the apple enzyme is not there sticking to this cuvette. Okay. So we are removing the previously used tips, and the next thing we will be pouring out here is our catechol, that is 60 microliter. So we are adjusting this to 60. We have taken a clean tip from here, and this we are taking this catechol solution, 60 microliter of catechol solution, and mixing properly out here. And the next important thing is there, we need to add 150 microliter of protein. So as I have discussed previously, this is quite crucial and should be done in a rapid manner, and but be careful, don't damage this cuvette or all the things while doing it in a hurried way. So here we have taken again this apple extract, and this is the cuvette. Here adding here, just 1 or 2 times we are mixing, we are putting in the compartment, closing it, and again start. So here we can once again we can change this one. And the upper limit also. Here we can see this absorbance increases once again. However, the value of this absorbance is quite higher from the previous one, since we can say the higher concentration of catechol was used here. But let us not be too sure out here, because we are bothered with the activity part and the activity value and the milliabsorbance per minute value. So the change in those values is actually what determines the rate of the reaction. For the timing, we can see that this curve is also similar to the previous one. The time out here is actually 300 seconds or 5 minutes. You can vary this time depending upon your convenience. Generally, a longer time period actually gives a better result. Okay, and in all the cases, a literature study have shown that higher time period gives a more accurate results in case of enzyme kinetics reaction. For the timing, let us discuss about this main table out here. So this one is the activity. So what is the activity? If we you have seen in your theory classes the activity of the enzyme. So what is basically the activity of the enzyme? Enzyme activity refers how enzyme is working. Well, activity depends on many factors. Activity depends on its substrate, activity depends on the type of enzyme, activity depends on the structural property of the enzyme, and many other factors out here. In this case, actually the activity depends on the substrate, because we are varying the substrate. But if we vary other parameters like temperature, pH, shall the activity change? Well, you can decide, because in the theory classes we have seen how the enzyme stability changes on pH or other temperature, and activity is no doubt related to the structural moiety of the protein. So, if there is any changes in the structure of the protein, the activity will surely change. Out here the parameters like temperature and pH are all same. Now, the pH we are using is 7.4, is pretty crucial out here, and we will discuss a bit about pH after a few minutes. For the time being, let us see how this curve goes. Again, it is attaining this almost flat shape out here, initially increasing and again this flat shape. It is 244, 245 and almost more or less around 50 seconds remaining. So, it is almost 275 seconds are there, and here we can see the increase in absorbance value is going, but however, the increase has slowly dropped, that is the rate of increase has dropped a bit after more or less 150 or 200 seconds, as we can see here. It is 295 and 5 more seconds. Okay. Well, we are done with this one, and again we have to save this. For that we have go to save as sample 2. Here again data printable. Now, the important thing out here is we have to note this too, the activity and the milliabsorbance per minute. Out here we can see the milliabsorbance per minute is around 83, and whereas the activity is 0.083. Okay. So, what happens is there both are actually same, the units are actually different out here. Okay. So, the milliabsorbance per minute or rather the activity, the rate of the reaction, as we can see, has increased when catechol has been added. So, again I am noting down these values. Now, once this experiment is done, we have to take out this cuvette, and if you take a closer look out here in this cuvette, although I am not sure whether this is visible or not, but you can see this is the dark brown color, or rather the yellow is very dark out here. Okay. So, it has been oxidized. Okay. So, now we have to wash this cuvette properly before proceeding into the next analysis. Now, we are done with 1 millimolar and 2 millimolar of catechol concentration. Catechol is used as substrate. Now, we will use 4 millimolar of catechol. For that, we will be taking the buffer as 330 microliter of buffer. We have set this pipette to 330 microliter, and now we will take this buffer. Here we have taken 330 microliter of buffer. After that, we have calculated the volume of catechol required. The volume of catechol required for 4 millimolar is 120 microliter. So, we have adjusted the micropipette to 120, and we are taking this fresh tip from this tip box, and this is the catechol solution. We are taking 120 microliter of catechol and we are adding out here and mixing it properly in phosphate buffer. After that, again the crucial step, we will be adding protein, that is 150 microliter of protein. We are opening this lid, taking a new tip and be adding this protein 1-2 times and immediately transferring it to the chamber, closing this lid and start. Again we have to adjust this one. Here we can see it is going upwards like this way. So from here what we can say is that this concentration is changing, the concentration of catechol in the mixture is changing and more of catechol oxidase is being formed. However, here we can see a small change in the volume or small change in the absorbance value, a certain fall. Actually these are some errors which generally happen while we are doing a particular experiment. But one thing you have to take in mind is that the milliabsorbance per minute value or the activity value is constantly changing, either increasing or decreasing with concentration of catechol. Generally, why such a fall takes place at moments? What happens is that the solution, sometimes the change or the substrate which is undergoing a change is formed at a particular place. Suppose in a cuvette, when there is a solution, it is being formed at a particular stage, in the middle stage. Okay, just assume it is being formed at a particular stage. Okay, and it is not actually being homogeneous. What happens is that when the light falls on that particular sample, what it sees is that it sees the concentration of the new product at that particular region, and as a result the concentration at that region is much higher as compared to others, and as a result the absorbance value is reported as much higher. But however, when there is actually a dissolution of that compound and the solution become homogeneous in that case, so what happens is that the absorbance value again drops because the concentration at that point again decreases and it becomes homogeneous. However, the absorbance value drops, the overall absorbance value, and however here we can see a sharp drop out here. And again sometimes bubbles are formed in the medium which might cause this change. So these are manual errors which are expected to happen once you start doing an experiment. So it is recommended often to use proper methods and proper precautions while doing this experiment. At times such a jump in the absorbance value might lead to an error in the result. In such cases we have to repeat it again in the same procedures out here. And in other cases what you can do, you can take a large number of samples, like what you have to do, you have to actually calculate the milliabsorbance per minute, that is the velocity. Okay, why it is velocity we are coming later on. So milliabsorbance per minute we have to calculate and we have to plot it in the y axis, and the x axis we have to plot the substrate concentration. So, at least you need 5 to 6 points. So, you can actually increase the number of data points to 8 or 9. Why? Because in some cases the data point for other, like for the second point, the third point might be filled with errors or something like that. You can actually omit those. So, here we can see it is becoming more or less smooth. And 15 seconds remaining. Now, what we have seen out here is that the value which comes is actually lower than the previous one, the milliabsorbance per minute. Okay, it's around 75, whereas the previous one is around 83. So, some changes or problem might have occurred in that region. As a result, we are repeating this one again. We have taken the same amount of phosphate buffer and catechol, and we are adding now 150 microliter of protein solution. Taken this protein, and now we are adding out here. And as soon as we have added, we are putting it here, close the lid and start. Now, here we can see this is again increasing. Okay. Unlike the previous one, this increment has started from this range actually. So, the previous one started from here, and now it started a bit lower than that. Sometimes it's happened like it first initially increases and the rate again gets slower, okay, and however again there is an increase. So, actually unless we plot the Vmax graph, we can't actually estimate or can't actually tell how the reaction is proceeding. However, the steepness for this curve seems to be a bit lower than that of the previous one. So, let us see how it proceeds. So, sometimes what are the chances of error in this reaction, in this procedure? See, while we are doing this experiment, the errors might occur because the time range for which you were giving this mixing the sample in the cuvette may not be same in all the 3 cases. Number 2, the enzyme what you are using out here, this enzyme, okay. So, it is being kept for a long time while you are doing this reaction. Generally, the protein which you use should be kept in a very cold condition, okay. So, the enzyme, there might be some changes in the enzyme or rather in this concentration of this entire volume, okay. So, such changes are subjected to giving a different value in case of our milliabsorbance per minute or the activity. Here again we can see it increases, but however it is coming close to this one. Now it is becoming almost smooth. Here we can see. If we extend this one a bit, we can see it is becoming smooth. Better visualization is obtained from here. This shows that the reaction might have started a bit earlier before we have pressed the start. These are certain changes, these are certain errors which might occur. So, the reaction must be repeated at least 2 to 3 times in cases, so that in order to omit the result which come from errors. It is already 260 seconds and 40 more, less than 40 seconds to go here. This looks more or less like Michaelis-Menten curve out here. The absorbance value has become more or less static, or rather it is changing very less. It is interesting, this plot is quite interesting. Here you can see this one, it first increased and again it is becoming linear. Actually this is the ideal plot. So, let us see how the value comes. Well, the value comes around 1, however, if we change a bit, generally we can change this part, this range, okay, and we can see how this value actually changes, the absorbance value. So, we can select a region where it is very steep. This region is quite steep, okay. So, this is basically around, it is less than 20, but it is okay in a possible region. So, it comes around 85. So, here we can see the constant, the milliabsorbance per value of the activity has actually increased. So, we have to again save this one as sample 3. Data text file, and we are again noting down the activity is 0.085, whereas the milliabsorbance per minute is 85.31. Previously it was 83, now it is 85.31. Now, what we can say is that in the previous one, this it showed a steep increase initially, that is why we get a much higher value in this case. So, let us move to the next concentration. Now, we are using 6 millimolar of catechol. For that, we have taken 180 microliter of catechol. Okay. We have initially filled this cuvette with buffer. We have taken around 270 microliter of buffer and 180 microliter of catechol. So, here we are taking again a clean tip of the catechol solution. Taken 180 microliter of catechol. You are mixing this one properly. And now the prime important thing. Now, before adding this protein, I would like to clean the sides of this cuvette, because after adding protein it is better not to do all those stuffs. Okay. So, now I will just open this lid with the apple extract, taking this one, taking close to this chamber, adding up down, putting it inside, and started it. Here we can see it's almost similar. Well, let's move that graph out here. Okay. So, here we can see this line, which we can't actually visualize properly out here. This is the orange colored line, and over this orange line here we can see another line is coming out here. This is basically here we can see this line is going from here. This is for our present sample. Here we can see properly, so easily out here, it's going upwards. Interestingly, what we can see is that it has initially increased a bit. If we compare this one minus 0.6, here it has initially increased a bit, but again it is decreasing. Well, what happens at higher concentration is that at higher concentration, this part is a flat part is basically the saturated part. Okay. So, saturation is happening in the enzyme. What does that means? That actually means that the enzyme, or rather this one, this catechol oxidase enzyme, the active side of the enzyme is getting occupied by this substrate. Now, as the substrate concentration is increasing, all the proteins are getting all the active enzymes, the active side of those enzymes are getting occupied. So, initially it is showing an activity, but however, since it is being saturated, all the active sites are being filled up by, or rather occupied. So, it is getting saturation at an early stage here. So, if we can compare the saturation from these two, we can see for the sample with around the blue line, as we can see the blue line. Here we can see the saturation is not that much prominent out here. It is getting saturated after 3 minutes or something, or rather 300 seconds or something like that, but however saturation is not coming out here. On the contrary, we can see as we have increased the concentration of this substrate, that is from 2 millimolar to 4 millimolar, this one is getting saturated out here, and from here we can see it is getting saturated almost around, if we could have drawn this a line out here, you could actually imagine it is getting saturated around 100 seconds. This one is the time, this is the second, around 100 seconds it has become saturated, or over than or more than a bit more than 100 seconds, that is the orange line. However, talking about this deep brown line, which is being operative currently for this sample for 6 millimolar, we can see saturation has been started before 100 seconds. So, it is an interesting thing. So, almost 235 seconds have gone, and it is hardly moving. Here we can see the absorbance value is becoming static, slightly moving, or it is more or less static, and we expect the velocity change should not be a bit significant. We do not expect it to be a significant change out here, because the upper one and the lower one more or less similar. So, it is 280 seconds and more than 280, around 15 seconds remaining. Two seconds, 1 second. Okay. So, now it is over. So, we can actually move this one and see in which range we get an optimum value. Okay. Out here we can see we are getting an absorbance, milliabsorbance per minute value of around 87. First let's save this one as sample 4. Enough text file sample 4. So, here we can see it is around 87. So, I am noting this down. For 6 millimolar for sample 4 it is 0.087 activity and the milliabsorbance per minute is 87.43. The previous one was 85 and this is 87. Now, we will proceed with 8 millimolar of catechol solution. Now, we will be dealing with catechol of concentration 8 millimolar. For that, we are taking 240 microliter of catechol. Okay. Now, for 240 we are taking actually 120 microliter 2 times, because this is 200 microliter maximum volume, 200 microliter micropipette. So, we are taking catechol 2 times, 1 we are adding here, we are mixing it. Well, I am using a fresh tip, you might not use it because the catechol out here is in phosphate buffer also, but here also in phosphate buffer. However, sometimes what happens actually protein might be there contaminated, so better to use a fresh tip. I am using here. The next thing I will be adding is protein, that is 150 microliter of protein. Okay, this one is the protein solution. Well, I have taken this protein solution out here. Before adding it, I am cleaning the sides of the cuvette. Now, I am just adding this protein solution gradually out here and then up and down 2 times and then start. Well, here we can see this one is increasing, this value should be minus 0.6, yeah, it is better. So, here it is increasing and let us see what happens. Well, it is increased a bit. I am talking about this part, the one in violet, here this one. Okay. I think we are again getting this interesting result. It has increased a bit, now it is becoming a bit smoother. It is gradually increasing, however, more or less what I can predict or what we can see from here is that it has become more or less flat, and now it is intersecting with the previous one. If we remove others from here, we can see that it has become almost in a plateau type of shape. Okay. And the interesting thing what we have seen in the earlier case, this saturation is becoming less than 100 seconds before 100 seconds. However, in here the saturation is coming, I mean at a value which is lower than this one, because here we can see at this point for the brown one, let us increase the resolution 0.4, okay, 0.4 minus 0.3, okay, fine. So, here we can see here at this point, okay, at the point where the this junction is occurring. At this point the saturation has started forming for the previous one, that is for this deep brown color, but however in this case the blue line or the violet line we can see the saturation has started somewhere around here, which is around 50 or something, 50 seconds or something. So, this shows that the saturation has being formed at an early stage when concentration is increased. So, we are predicting for like 4 millimolar or 6 millimolar the saturation is coming, and again for 4 millimolar let us see what it is. Here we can see the saturation for 4 millimolar is something at this point, for 6 millimolar it comes around this point, 4 millimolar is over 100 seconds basically in a quantitative way, over 100 seconds. For 6 millimolar it is just less than 100 seconds. Okay. However, for this 8 millimolar what we are getting, it is around 50 seconds. Here we can clearly see this one. So, this is quite sure that what actually happens is that protein are there in the medium or the enzymes are there which can actually cleave or catalytically oxidize this catechol substrate. Okay. Now, what happens is that if the catechol substrate is increasing, okay, or if the catechol substrate is increasing, then all the active sites are getting blocked, as I have discussed, or rather all the active sites not blocked, all the active sites are getting saturated, and as a result the saturation is coming at an early stage. So, as the concentration is increasing, more and more enzymes are getting saturated. So, the number of free enzymes are decreasing, as a result the saturation is coming at an early stage. Now, what we can do is that we have started with 1 millimolar. So, we can actually shift to 0.5 millimolar. Okay. So, we can see how this comes for 0.5 millimolar. So, it is 268, it is coming 270. Now, regarding pH, what I was talking is that in case of pH, when pH changes, okay, we are doing it around pH 7.4. So, if pH changes, pH increases or decreases, it might affect the structure of the protein and thereby its enzymatic activity. Well, it is 300 seconds is there, and we can actually regulate this. We can actually alter or rather select a particular concentration, a particular range where we have to actually select the activity. Well, the activity at this range is coming around more or less around 89. Okay. So, I am noting this down for 8 millimolar 0.0897 and 89.668. Now, I am doing this with 0.5 millimolar of catechol. Now, although we have started with 1 millimolar of catechol, however, out here we are taking 0.5 millimolar catechol. The concentration of catechol here is 0.5 millimolar, and for that we are taking 15 microliter of catechol sample. Now, this is a 20 microliter pipette and we have adjusted it to 15 microliter. Now, I am taking this one, adding this 15 microliter of catechol, and after that I will be taking this protein once again. I am cleaning the side of the cuvette first, then keeping this lid opened and ready. Okay. Now, I am adding this protein carefully, add this one, and now start. .6 and it is minus 0.2. We have completed the kinetic analysis, that is the enzyme kinetic study of catechol oxidase, or rather the apple extract along with catechol. Okay. So, now what we need to do is that we need to add one inhibitor and see how our this milliabsorbance per minute value or the activity value changes. As inhibitor, we know that inhibitor actually blocks the active site of the enzyme and the substrate, whereas couldn't actually attach with the active site. Basically, depending upon the competitive or non-competitive inhibitor, the substrate attaches accordingly, or rather the inhibitor attaches accordingly. Okay. So, if inhibitor attaches to active site with increasing concentration of substrate, in case of competitive inhibitor, we can get the Vmax, as we have seen from the theory classes. For that, what we can do is actually, we can actually change our experimental procedure. Okay. So, how to change the experimental procedure? One thing we can do is that we can just change the way in which we are adding the individual constituents. For example, initially we are adding phosphate buffer, then catechol, and after that protein. Now, in doing so, what we are actually doing is that the protein is acting on catechol. Now, we can proceed with this experiment in two ways. Number one, you are adding phosphate buffer, then catechol, and then we are adding the enzyme, or rather the apple extract. And number two is that we are taking the phosphate buffer, then adding the enzyme, and then adding the inhibitor. What is the difference in two cases? Basically, what happens is that when you are keeping enzyme and inhibitor, or rather enzyme, the apple extract, and inhibitor, that is we are using here para-hydroxybenzoic acid in the cuvette, what actually happening is the inhibitor is attaching to the active site of the enzyme. And now, when we are adding substrate out there, that is the substrate means our catechol. So, what is happening is that it cannot actually get into the active site at that moment. However, if you are increasing the substrate, it might go into the active site if it is competitive. If it is non-competitive or uncompetitive, some other phenomenon might take place. On the other way, what happens if we add this inhibitor and catechol both at the same time in the cuvette, and then enzyme we are adding. So, there might be a competition whether which one will be added, the enzyme, the substrate, or the inhibitor. So, there might be a competition. Both processes are equally accepted, and all the experimental data which are carried out in this research activities, both processes have been tried out. There are sometimes failures, sometimes some positive feedback from all these procedures. Now, in this case, what we are going to see, we are going to take the normal procedure. What is the normal procedure? We are taking the enzyme, we are adding the inhibitor to it, and we are keeping it, and after that we are adding the substrate. See, the result may come or may not come in this way, but before you approach with any inhibitor, you have to try two ways. Whether it comes in the first way or in the second way will depend upon the plot. In our case, we will go with the first way, in which we are taking this cuvette. Okay. In this cuvette, we will be filling the phosphate buffer first, and after phosphate buffer we will be adding enzyme, and after enzyme we will be adding the inhibitor, that is para-hydroxybenzoic acid, and after that we will be adding catechol. Okay. So, let us proceed with the analysis. Okay, the concentration of inhibitor we are keeping here is around 0.8 millimolar, or it is around 1 millimolar or something like that. So, it depends upon the concentration of inhibitor. Outer the inhibitor is around 0.8 millimolar, and for that we will be taking 50 microliter of inhibitor. But before that, we have to take the phosphate buffer. The phosphate buffer we will be taking out here is actually 370 microliter, and the catechol concentration out here is 1 millimolar, like the previous experiment. Now I have washed this cuvette properly and I am taking the phosphate buffer. It is 370 microliter. Then I will be adding the enzyme. It is 150 microliter of this enzyme. I will be adding here. So, until and unless you add the substrate catechol, the reaction won't start theoretically. Okay. Now, I will be adding 50 microliter of inhibitor, that is para-hydroxybenzoic acid or PHBA. Inhibitor, and I am adding 50 microliter of it. Two things you can do after adding. You can keep it for incubation for 1 to 2 minutes, or you can directly add catechol to it. Whatever you do, fix this time gap same for all the readings, that is with increment in catechol concentration. Now after that, we are using the same micropipette with a different tip, of course, for taking catechol. We will be taking 30 microliter of catechol. Now out here we have to start as soon as we add catechol. So we are opening this lid, taking catechol here, mixing it and start. Well, here we can see the absorbance value is increasing. The main thing we have to consider out here is the milliabsorbance per minute, and this is actually the velocity. Now, when we are plotting this experiment, so what we are doing, you are taking the velocity in the y axis and substrate concentration in the x axis. For plotting Michaelis-Menten equation, from where you can get the Vmax value and the Km value. Okay, the Km value is better from Lineweaver-Bach plot. However, Vmax value you can get from here, and for that what you will be doing is that you are in the velocity. So what is velocity? Velocity is the rate of a reaction, as we know. And what is the rate of the reaction? It is actually, rate is the change in concentration with time. So a rate means with time. So rate of reaction means change in concentration with time. However, here we are not measuring the concentration, we are measuring the absorbance. So why change in absorbance is referred to as rate? Because as we know from Lambert-Zwiat law that absorbance depends on concentration. Okay, A equal to epsilon into C into L. So A, that is the absorbance, is directly proportional to the concentration. So the change in absorbance basically indicates the change in concentration. So the absorbance, for taking the rate, we can take C2 minus C1, that is the concentration for at time 2 minus the concentration at time 1 divided by T2 by T1. Instead of C1 and C2, here we can take A2 and A1, that is A2 is actually the absorbance at time 2 and A1 is the absorbance at time 1, and this is actually what is giving milliabsorbance per minute at this time is actually the velocity, that is the change in absorbance with time. Now apart from that, what you can do for this is that this value is being provided by the software. But if you want to wish to get a much better results, what you can do is that you can take a particular time range from here and you can actually subtract the absorbance manually from the text file. You can get the absorbance for a particular time, you can actually subtract it, and for the change in absorbance, the change in time, you can also do it manually. What previously in previous instruments which did not have such facilities, they used to do it in this way, where they manually used to plot it, and from there they used to take those values and calculate the velocity. Now, here we can see this is increasing with time, which indicates that there is a change in the concentration of the component which is being formed. Okay. So, actually here it is increasing, this means the component is being formed, that is the oxidation is being promoted more and more, and the enzyme is acting on it. So, catechol is being oxidized to benzoic acid, or rather parabenzoquinone, not benzoic acid, it is parabenzoquinone, or basically for catechol it is just normal benzoquinone, orthobenzoquinone for catechol. Okay. So, in case of benzoquinone, what happens is that it absorbs at a higher wavelength, okay, around 540, and this the more benzoquinone is being formed in the medium, the more of absorbance increases, and from here we can get this increase in absorbance value. So, almost 250 seconds have passed. And see here, we can't see that any significant saturation in this case. However, the value is not that much increasing, saturation might be coming out here because the change in absorbance is not that much prominent in this case. Now, we have to see what the activity data shows. Well, interestingly, we can see here the milliabsorbance per minute reported out here is 34, okay, which was in case without inhibitor it was 57. Okay. So, what we can see is that in presence of inhibitor, the activity or the rate has decreased. Here you can see the activity, it is 0.0342, and here the activity is 0.05. Okay. Of course, 0.05 for the enzyme was much greater without inhibitor, whereas in presence of inhibitor has decreased to 0.03. So, again we are noting down this value. In presence of inhibitor with PHA parahydrobenzoic acid for serial number 1, we are getting an activity of around 0.0342 and milliabsorbance per minute, which is 34.21. Now, we will keep the concentration of inhibitor same out here, and we will vary the concentration of substrate, because when we are plotting the change in the concentration of substrate. Sometimes people might do it by mistake, instead of changing the concentration of substrate, they start changing the concentration of inhibitor, but never do that. Always we are bothered with the rate at a particular concentration of the inhibitor. So, out here the concentration is 0.83 millimolar, and we are changing the concentration of catechol now. Next, we are taking 2 millimolar of catechol. For that, we will be taking 340 microliter of this buffer, phosphate buffer 340 microliter, and taking in this cuvette. And now the amount of inhibitor, it is PHBA, is 50 microliter. So, it should be same, but before that we have to add actually the protein, and is 150 microliter. We have taken 150 out here, and this is the protein, and adding 150 microliter. After that, we are adding this inhibitor. (50 microliter of inhibitor), and kindly mix it carefully. And after that, what we are doing? We are adding catechol, and the volume of catechol is 60 microliter. So, before adding catechol, once again what we should do, should keep this lid open, wash the sides of this cuvette prior to the addition, and take catechol and bring this close to this sample compartment, add this one, -0.6. Well, here we can see it has started increasing, but the value is a bit lower than out here. But as we know, as we have said earlier, that in the absorbance might start increasing from a lower value or higher value depending on when you are keeping the sample, because at all the time at the same time we are not keeping it here. So, the thing we should be concerned is the milliabsorbance per minute value. So, let us see what it comes. Now, we can see here this one is increasing a bit, and it is coming close to the previous one, the previous line. Generally, what happens is that when inhibitor occupy the active site of the enzyme, it either allows the substrate to bind with it, may not allow the substrate to bind with it, or sometimes it may happen it changes the conformation of the protein. Okay. So, depending upon the, if the conformation of the protein changes, so what happens basically, the substrate might not bind with it anymore, and such type of inhibitors are generally referred to as suicide inhibitor. Okay. So, why? Because they bind with the protein in such a way, these are not the suicide inhibitor, they form a covalent linkage with the protein, okay, and they bind it, they actually block the protein activity. So, sometimes what happens is that neurotransmitter, some neurotransmitter, or these are enzymes, or there is some enzyme during the neurotransmitter and all those. So, if any types of poisonous gas comes within or rather attaches to those enzymes, what happens is that it blocks those enzyme, and as a result our neuron fails to function, or rather body other activity fails to function, which leads to fatal outcomes in our body, and sometimes people might not even continue living in such cases. So, what happens with inhibitors are sometimes good, sometimes bad. Inhibitors are good in cases when they are used as drugs. Okay. So, what actually is drugs are something which blocks the malfunction of an enzyme. Suppose an enzyme is acting in such a way in which it should not act, or rather it is showing some excess of its function, like an enzyme should have an optimal activity. If excess of function is showing, then it is also harmful. If it is lower activity is showing, it is also harmful. Suppose an enzyme shows an activity which is very higher, which is not desired actually. Okay. In such cases, what happens is that you should need something which will block those enzymes, and in that cases inhibitors are promoted in the form of drugs. Okay. So, in our body, I have also discussed a bit of this in the previously. In our body, what happens if any diseases is there, it might be sometimes due to malfunction of some enzyme. So, drugs are promoted which actually blocks those enzymes. In doing so, what happens is that drugs might actually interfere with other enzymes. Okay. So, as a result, other enzymes which are not actually targeted, the drugs are not targeting that enzymes, they might not function properly. As a result, what happens when we take too much of antibiotics or drugs, it generally hampers our body function, regular function. Okay. So, it is advised not to take so much drugs because ultimately they are inhibitor of enzyme, and all the inhibitors are not that much specific in cases. So, here it goes around 300 seconds. Well, interestingly, again we can see the activity it is showing here is 53. As I said here, it should be, it is higher, but however it is going lower than this red line, and it is not a big issue. We are concerned with the milliabsorbance per minute activity. It is showing 53. However, for the same concentration of catechol without inhibitor, it was actually 83. Okay. So, we are noting down the milliabsorbance value 0.053 and it is 53.834 milliabsorbance per value. Now, we will move with 4 millimolar of our catechol. Okay, now, we are taking this catechol of 6 millimolar concentration, or rather 4 millimolar concentration. For that, we are taking 280 microliter of buffer, and then protein we are adding 150 microliter, or rather the enzyme, and now we will be adding 50 microliter of our inhibitor. That is para-hydroxybenzoic acid. We have mixed it properly, and the catechol we require out here is actually 120 microliter. So, we are fixing it to 120. And before adding catechol, I have just opened this one. Taken this catechol 120, I am adding here 1, 2 times, done. Close this lid and start. Here we can see a green line, a faint green line is appearing, rather, and so in this way, this green line is appearing, which is for the absorbance, change in absorbance with time. Basically, we have to go on like this for a particular concentration of substrate. You have to add a particular concentration of the inhibitor. Now, it is better not to take the substrate concentration which you have not used without inhibitor part, because in the first part, suppose I have taken 1 millimolar, 2 millimolar, 4 millimolar catechol, and for inhibitor, when we are taking with inhibitor, we are taking 1 millimolar, 3 millimolar, 6 millimolar or 5 millimolar catechol, which was not there in the first set. So, what happens in this case is that you cannot actually properly correlate. Well, you can get a proper experimental data also, but you can't actually correlate like the way we are doing out here. It is better to take those concentration of the substrate in presence of inhibitor, which you have taken earlier without inhibitor. So, here we can see there is a small fluctuations out here. Basically, what happens is that this experiment is good. Why? Because we are actually seeing something which we regularly observe, but we do not, or rather cannot quantify, because we never ask anyone that okay, you are seeing apple is getting oxidized and turn brownish. tell me the rate at which it is getting oxidized. Well, people won't answer you such questions also, and because they don't have this UV setup, well, you can actually get answer by yourself by doing this simple experiment, where you don't have to buy expensive enzymes or expensive protein, or this catechol is also very easily available, and you can actually get this apple extract in the easy way, and it looks good basically. However, some drawbacks of this experiment, what you can actually think of is that in a particular fruit there isn't only one type of protein, or rather one type of enzyme in the apple extract. There may be multiple enzymes are there in a fruit. Okay. So, the action of different enzymes on this substrate is not at all evaluated in this case. What we are getting the result is the combination of different enzymes on catechol. It is not the combination of only catechol oxidase on this enzyme. Okay. Number 1. And number 2 is that we do not know the concentration of catechol oxidase out here, because we have taken a particular amount of apple from this case. So, we need to actually quantify the amount of protein we are adding. When you will be doing any enzyme kinetics with whatever enzymes in a phenylalbumine or phenylribonuclease, you need to know the concentration of the enzyme. Okay. So, concentration of enzyme is very much required out here. These are few of the drawbacks, and there is another drawback out here is that when you are adding this, or rather I am not telling about this experiment, where the entire kinetics step is that when you are adding all this, you have to completely take a monitor of the time you are adding. So, manual error might occur. It is expected that manual error might take place in this way. So, in order to avoid those manual error, some other techniques should be introduced in order to study the kinetics. Okay. So, here we can see saturation has started occurring. Here it is coming. So, this one for sample 3. Okay. This orange color was for sample 3. I did not actually save the others, so you can't actually see here. Now, it is coming here, it is much lower, the stiffness is much lower than that of the sample where catechol was present without any inhibitor. This one was without inhibitor, and this one is with inhibitor. Well, out here we can see the rate is increased to 79, or rather activity point 079, and the milliabsorbance per minute is 79. So, the milliabsorbance per value is again increased. However, in the previous without inhibitor the value was 85, as I have noted previously. Here the value is 0.079 and it is 79.65. The activity is gradually increasing with increase in concentration of catechol. However, this is not the same as that where inhibitor was absent. That rate was much higher. Now, let us proceed and see what happens for 6 millimolar of catechol concentration. In this case, what we have seen is that it goes to a value of around 80.58, the milliabsorbance per minute value. So, in the previously it was 87, as we have seen earlier it was 87, now 0.086 and it is 80.58. Actually, in this way what we can do is that we can actually go on increasing the concentration of the substrate, and from there what we can say is that we can get the V value. Now, these milliabsorbance per minute value are the V, that is the velocity. Okay. So, we need to do is that we need to take those velocity and plot it in the y axis for a particular concentration of substrate. We will get something like this. Initially, as we have seen in this data, it started from 57 and it gone high to 83 or something like that, then 85, and then 87, and almost it is becoming linear like the Michaelis-Menten. Similarly, in this way, it started from 34 without inhibitor and then went to 53 from the data I am saying, and then 79 and then 80, and now again it is decreasing. So, well, once we plot this graph at very high concentration of substrate, we can see whether it reaches the Vmax or not. From there we can calculate whether it is comparative or non-comparative inhibitor. Well, so what we have seen in this experiment, we have studied how to carry out enzyme kinetics reaction. Initially we have prepared catechol solution, para-hydroxybenzoic acid solution, and phenylthiourea. Well, phenylthiourea we can use also and see how the kinetics changes. We can actually compare the two different rates and see whether for a particular concentration of catechol which gives higher velocity, for phenylthiourea or for para-hydroxybenzoic acid. If the V value or the rate is higher for phenylthiourea, that means para-hydroxybenzoic acid is a better inhibitor since it decreases the rate. In this way you can compare. So, it is always better to use two different types of inhibitor. Secondly, we have extracted the juice from the apple by grinding it in a mortar pestle, and we have used it, we have actually filtered it using a cloth paper. Sometimes you can actually centrifuge it and we can use the filtrate from there, and however we have seen that it is almost a clear solution. So, we can easily use that filtrate, and you have used it as a source of protein. Initially we have taken the phosphate buffer pH 7.4 and 10 millimolar. You can use 20 millimolar also, and that phosphate buffer, what we have done is that from phosphate buffer we have taken the phosphate buffer we have taken in a cuvette, 10 millimolar of pH 7.4 phosphate buffer, and to it we have added initially catechol and then protein and immediately given it for scanning. Now, in case of inhibitor, what we have done, we have taken the buffer, added protein first, then inhibitor, and then we have taken it close to the instrument and added catechol at that moment. So, I am not telling this is the ideal way of doing the reaction. You can do it other way. You can add enzyme, and like before adding enzyme you can add the inhibitor and catechol. You can see it yourself. Once you start doing the reactions by yourself, you can apply whatever method you think will be best. For the time being, I hope this was quite fruitful, and how to carry out with enzyme kinetics the basic step we have learnt. Thank you.